Good afternoon. My name is Brody Holland. I'm going to be presenting about sickle cell disease. Here's my general outline for today's presentation. I'm going to cover the introduction, background, statistics, molecular cause of the disease, the mode of inheritance, genetic testing available for the disease, famous people with sickle cell disease, and the conclusion. It is important to learn about different genetic disorders, how to test for them, and how to treat them. If left untreated, sickle cell disease can lead to a shortage of red blood cells, causing fatigue and ultimately damaging blood vessels and vital organs. It has the highest prevalence rate among inherited blood disorders in the United States, and it can lead to anemia, which is a shortage of red blood cells, and can cause severe pain if left untreated. Sickle cell disease is an inherited red blood cell disorder that affects how hemoglobin is produced. Because hemoglobin is not produced correctly, sickle-shaped cells instead of circular red blood cells are formed, leading to all sorts of problems. Roughly 100,000 Americans live with sickle cell disease every day. 1 out of every 365 African American births have sickle cell disease. 1 out of every 16,000 Hispanic American births have sickle cell disease. And every 1 out of every 13 African American babies are born with sickle cell trait. This does not mean that they have sickle cell disease, but they have one copy of the disease. The hemoglobin subunit beta gene mutation is responsible for the onset of sickle cell disease. The hemoglobin subunit beta gene is found on chromosome 11 on the p15 band. Hemoglobin subunit beta and other protein subunits provide instructions to the body on how to produce hemoglobin. Due to a genetic mutation, the instruction process is not complete and sickle-shaped red blood cells form instead of circular-shaped red blood cells. In people without sickle cell disease, hemoglobin instructions were sent correctly and the resulting red blood cells are circular-shaped, as you can see on the picture below on the left-hand side. In people with sickle cell disease, the instructions were sent incorrectly, leading to sickle-shaped red blood cells. These cells lead to blockages in the blood vessels, resulting in lasting damage and severe pain in the body if left untreated. In the picture, you can see the sickle-shaped red blood cells and how they can form a blockage in a blood vessel if left untreated. The mode of inheritance for sickle cell disease is an autosomal recessive pattern. This means that in order to inherit sickle cell disease, both parents must pass a copy of the mutated hemoglobin subunit beta gene. It is possible for a carrier to have sickle cell trait their entire lifetime and never develop sickle cell disease. In this pedigree chart example, both of the parents are carriers and neither of them have the disease. However, in their first generation, their first kid on the very left, the blue circle, is a female. There is their female child who does not have sickle cell trait or sickle cell disease. Their second child is the blue box in the middle, and he does not have sickle cell disease or the trait as well. Their next child is a female, and it is shaded half blue, half red. This means that the female is a carrier for the trait, but does not have sickle cell disease. Their next child, however, as shown as the red shaded box, is a male, and he has developed sickle cell disease. This means that both of his parents passed down the mutated genetic copy. There are several genetic testing available for sickle cell disease. Amniocentesis uses a small needle to obtain a sample of amniotic fluid from the mother. The test is able to tell if the fetus will be a carrier or have sickle cell disease before it is even born. Pre-implantation genetic diagnosis allows parents who are carriers to test their embryos before they conceive. They can choose to only implant embryos that do not carry the disease. Blood tests check for the hemoglobin S, the mutated form of hemoglobin. If this test comes back positive, doctors typically order hemoglobin electrophoresis, which is another blood test that can look at different forms of hemoglobin in the blood. What age should you get tested? All newborn babies should get screened for sickle cell disease as early as 24 to 48 hours after being born. Is early detection important? Early detection is important because it can help manage the disease more effectively and reduce mortality rate. All newborns must be tested in the United States by law. There are many famous people who have lived with or are currently living with sickle cell disease. Here are just a couple examples that I have picked out to show you. Tiki Barber was an NFL running back for the New York Giants. He had a very productive career despite his challenges with sickle cell anemia. Miles Davis was considered one of the greatest jazz artists of all time. He pushed the envelope of what was known for jazz and inspired many others. For treatments for sickle cell disease, the only FDA-approved treatment is a bone marrow or stem cell transplant. This is a procedure that is formed that takes healthy cells that form blood from the donor to the recipient. It has many risks, and the donor must be almost identical match for the transplant to work. It is typically reserved only for children with severe sickle cell disease who have minimal organ damage. For my conclusion, sickle cell disease is important to learn about. Early testing and treatment is essential to improve the quality of life with people who have sickle cell disease. More research is needed for further treatments. And because it is so hard to find a match for bone marrow and stem cell donors, many donors are needed to change lives. Here are my references. Thank you.